Thank you, NAPB, for inviting me to present today. My name is Nicolas Morales, and I'm a fourth-year PhD student majoring in plant breeding and genetics. I'm also minoring in statistics with a focus on data science, as well as minoring in environmental information science. My co-advisors are Dr. Kelly Robbins and Dr. Lucas Mueller. Today, I will present my research detecting genetic and local environmental effects using high-throughput aerial imagery. My research aims are essentially twofold. First, I want to understand the genotype by environment, or G by E, effects present in the collected aerial image high-throughput phenotypes. Specifically, I'm looking at G by E originating from local field effects, such as from soil heterogeneity, from soil elevation, soil curvature, or composition. As we collect more data across years and field locations, we can then look at G by E across these environments. The high-throughput phenotypes I'm currently looking at are simple vegetation indices, such as NDVI. However, from the collected aerial images, we can potentially extract more informative and predictive features other than vegetation indices. NDVI was a good starting point given research indicating it is a heritable phenotype indicative of plant health. Secondly, I aim to monitor the stability of those high-throughput phenotypes for genotypes across environments and across the growing season. I intend to use reaction norm models to look at genotypic deviations across environments in the growing season. To probe these questions, I'm flying an unoccupied aerial vehicle, or UAV, mounted with a Mica Sense 5-channel multispectral camera. This camera captures images in the blue, green, red, far red, and near infrared spectra, as you can see here on the right. It has been a great learning experience working the logistics of flying throughout the growing season, from getting an FAA license to transporting and maintaining the UAV. The UAV flies a pre-programmed path over the field at an elevation of 25 to 35 meters. I'm flying over hybrid maize field experiments in the Genomes to Fields program over the past 3 years now. I'm also flying over alfalfa field experiments, but I won't show this here today. I try to fly twice per week over the entire course of the growing season. Then high-throughput phenotypes are extracted for each plot for each time point. ImageBreed is a web database software I've developed that allows drawing plot polygon templates, such as the one shown on the right in blue and red. The plot images are then saved along with the extracted high-throughput phenotypes. I'm very proud of the ImageBreed tools because they are an end-to-end platform for processing aerial imagery for plant breeding. Images coming off the camera can be directly uploaded to ImageBreed and high-throughput phenotypes extracted for each plot. ImageBreed builds on BreedBase, which is a database we developed in Lucas Mueller's group and is currently still in development. This first publication roughly describes the aerial image tools and processing, and this second publication describes in detail how the high density genotyping data is stored in the system. In short, ImageBreed connects the aerial images and their high-throughput phenotypes to the field experiment and to the high-density genotype data for the hybrids. As part of the Genomes to Fields program, we have high-density genomic data giving close to 1 million marker genotypes for the maize hybrids we've tested. And this is loaded into ImageBreed, allowing genomic relationship matrices to be calculated. To illustrate, here we can see 6 time points flown over a hybrid maize experiment in 2019. This experiment was 612 hybrids planted over 800 plots. Again, in blue and red, you can see the plot polygons that were assigned in ImageBreed. Vegetation indices were extracted, namely NDVI, for each plot and for each time point. The collected high-throughput phenotypes can then allow us to see the performance of different hybrids over the growing season. This chart shows mean NDVI values for 15 maize hybrids across 12 time points. We can see trends across emergence, flowering, and ultimately senescence. We can see NDVI shows stable trends for the hybrids, giving us assurances in our aerial imaging methods. My research is then separating genetic effects from environmental effects present in those high-throughput phenotypes using linear mixed models. The random additive genetic effects are modeled using genomic relationships among the hybrids, while the random environmental effects are found using spatial corrections and random regressions. To model the spatial correction, I'm looking at two-dimensional splines as well as separable autoregressive processes, or AR1 by AR1. These spatial effects draw information based on the row and column positions of the plots in the field. The random regression is distinct from the spatial corrections in that it draws information from the repeated nature of the phenotype over the growing season. The random regression has computational advantages over the spatial correction models and allows computing a continuous covariance function across the entire growing season. The random regression also allows more flexibility in defining the covariance structure between plots. The results from all of these linear mixed models are of the same form, and here I am showing representative figures. They give the additive genetic effects for each hybrid across time, as shown on the left, where each line is a hybrid across time, and they give the local environmental effects for each plot across time, as shown on the right. Each one of these heat maps is the environmental effect present across the field at a given time point, and I'm showing 6 time points. Here we can consistently identify a poorly performing region near the middle of the field, for instance. Given that these linear mixed models are essentially unsupervised and that we don't know the true genetic or environmental effects present in the high throughput phenotypes, I'm using 2 approaches to compare the models. The first approach is to use simulation and involves simulating an environmental effect across the time points and then seeing how well the linear mixed models can recover that effect. Here on the left, we see a linearly simulated effect that is constant through time over the 6 time points. On the right, we can see how well various tested linear mixed models can recover that effect. With this showing that the prediction accuracy of various models and scenarios. We can see in this case that the separable autoregressive process and the 2-dimensional spline process can recover this effect well. Here we see a second simulation where the simulated effect follows a more complex gradient and is constant through time. On the right, we see that the random regression does well at recovering this effect. There are many ways to simulate, and I'm looking at 6 different processes. So I'm not showing the 4 other processes here. My results indicate that specific linear mixed models are better than others at recovering specific types of effects. In other words, not all linear mixed models are created equally. And so aerial imagery may allow us to classify a given field based on the effects present in that field. Thereby, we can know the best type of linear mixed model to use to account for the local environmental effects and to make better genotypic selections. The second approach for comparing these linear mixed models is to compare with soil data we collected. So we conducted an EM38 soil survey in 2019 and in 2021. And here I'm showing the 2019 data. So you can see on the left, soil electrical conductance, or EC, and then on the right, soil elevation or altitude. And and these values were Kriging interpolated over the field from the soil survey. I then calculated numerical derivatives of the EC and altitude to look at curvature in the field. So DEC is the first derivative of EC, while D alt is the first derivative of altitude. And these are numerical two-dimensional derivatives. And these results indicate that soil elevation curvature explains the linear mixed model environmental effects well, but there's more research to be done. And I would like to thank everyone in the Robbins and Mueller labs, as well as my funding sources. And if you have any questions, please feel free to contact me. Thank you.